Good afternoon. I'm going to talk about genomics aiding pandemic research and specifically COVID-19. This work was carried out by Guy Shapira with the help of Eden and Roni from the lab and in collaboration with Professor David Gurevich, KSM Maccabi Center and Shamir Medical Center. In the lab, we work on genomics, so we combine biology, computational biology or bioinformatics together with clinical questions. And the aim is to better understand the disease and to reach better decisions next to the bedside. We are basing our work on 3 revolutions that actually started about 20 years ago. The first reading of the human DNA from beginning to end was in the year 2000 or 2001. And then throughout the years, we're trying to understand, we and many other researchers around the world, what do mutations or changes in the DNA mean? And we all know that in 2020, after reading the DNA, understanding it, we all walk around with mRNA in our shoulder, so we can also write the DNA or RNA and deliver it to people. We also know that this is based on advanced in machines, so NGS, next generation sequencing, which we use a lot, in accumulating a lot of data, a lot of bioinformatics, computational power to understand these changes. So we are basing our studies on revolutions that carried out in the past 20 years. I'm going to talk about multiple levels of research in our lab. Some of it is only hypotheses that we're testing now, and others are more advanced. But we do study DNA, RNA, proteins, antibodies, and also electronic medical records, and we try to combine them all. This is not our study. This is a study published last year, and everybody was trying to explain how come some patients die very rapidly from COVID-19, some do not suffer from COVID-19, and you could also see it on a global level in different countries. So here you can see a plot of strict rules in particular countries and cultural tightness and obedience of that country in following these rules. So you can see this graph, they made this correlation where there's more strict rules and more obedience, there's less mortality. This is what their claim, and if you want to look for Israel, just look at the top left-hand corner. Less cultural tightness, less obedience, and more mortality. But of course, we'll answer questions at the end. But of course, we're looking at other features. So we were scanning and trying to understand, is there a DNA basis for these changes? And we were looking at many different DNAs and mutations and polymorphisms. And this is one of them, one gene, AAT, alpha-1 antitrypsin, which is a protein that protects the lungs and the liver from damage. There were many studies carried out after 9/11 in the US, where many firefighters went into a building with a lot of smoke and they came out, and half of them suffered from severe diseases and others did not. So this was pointed out that mutations or deficiencies in this particular gene can cause protection or deprotection and vulnerability of specific firefighters in this case. We wanted to plot this tens of millions of people around the world in particular countries and the different ethnicities and the different mutations or polymorphisms in these genes versus their mortality in that country. And you can see the plot we generated here. So we took very large countries where we have a lot of genetic information, genetic data. And in this plot, we have a hypothesis that this might be involved in some vulnerability of some countries, some ethnicities for the disease. We're now testing it in larger cohorts that we're collecting here in Israel. So this is a hypothesis about the DNA. We have hypotheses about RNA. So here we downloaded different data sets of RNA-seq, upregulated and downregulated RNAs in nasopharyngeal swabs. Now it is known that one of the clinical features of infection with COVID-19 is loss of smell. So here you can see the olfactory receptor. You can see active on the right-hand side and inactive on the left-hand side. And with upregulation of a protein, of a set of proteins, RGS, we have a shift from active to non-active. When looking at the different data sets, we saw this protein, the regulator of G protein signaling 2, upregulated in positive infected versus negative in 2 independent sets of patients in swabs in the RNA expression. We wanted to expand this and look at other genes as well. We were looking for co-expressed genes. We found another gene, you can see it on the right-hand side, co-expressed with this gene with RGS2. We were scanning this. We're looking also at other sets of microRNAs. We know that microRNAs control these genes. Now we're building a network of regulation of gene expression that could be associated with the loss of smell upon infection with COVID-19. So our idea now is that a microRNA goes down. This specific microRNA was shown to regulate these two genes. When it goes down, these two genes go up, and an active to non-active receptor, olfaction receptor, actually takes place. When looking at antibodies or looking at proteins, we have a very large study with Shamir Medical Center. Over there we work with Dr. Adina Bar-Chaim and her team. They have been collecting or running tests or assays of serology of proteins. Now I'm showing you about 30,000 tests they carried out in their clinic. And you can see here on the left-hand side is naive, non-infected with a virus and non-vaccinated. This is the level, an assay of the spike protein, the level of antibodies against the spike protein. In the center here, you can see the level of antibodies against spike protein of infected individuals. And on the right-hand side, you could see those vaccinated 3 weeks after the vaccination. You can see a very large difference between, for example, vaccinated on the right-hand side and infected in the center. Of course, I stress out that these are antibodies only against the spike protein. If you think about it, if you're infected with a virus, you probably have a multitude of antibodies, not only against the spike, so the effect would be different. But if you're looking only at antibodies against the spike, this is the multitude. We're plotting it against age. Here in the center, you can see the age is 50. On the left-hand side, you could see 20, and about 80 and 90, you can see on the right-hand side. This is 3 weeks after the second vaccination. That means that if you're looking at the effect, the average effect of a 60-year-old 3 weeks after the second vaccination, it's about 30% less than if you were at the age of 20. And it's known already that the effect of the immune system is different for older age when comparing to younger age. If you're looking at the infection, the levels of antibodies against the spike during infection, that's the light blue line beneath, we can see that there's a dip at ages 20, 30, and 40. We can't explain that yet, and you can see a higher level at older ages. We also saw differences between males, the effect of males and females, antibody levels against spike. When comparing the two, this is the infection, and we can also see the difference between those suffering from symptoms in red and those that did not have any severe or strong symptoms. You can see that in green. We can also see a divergence between males and females upon effect with and without symptoms. We see the divergence here for women at the age of 50 or 51, and for males at the age of 30 or 35. When discussing this with the physicians, we believe that the effect here is based on the change in hormonal expressions in the body. And what we're doing now, again with Shamir Medical Center, is every time they're taking these serology tests, they are also running assays for hormone levels. And we're trying to find out the correlation between hormone levels and the antibody effect. By the way, generally, the levels of the immune system is stronger in women than in male. This is also a known phenomena. We're looking at electronic medical records, and this study was carried out by Ronnie in the lab, and this is a collaboration with the Maccabi Research Center with Dr. Sivan Gazit and Dr. Tal Patalon. We were seeking for Teflons, meaning those that were exposed to the virus, but are not carriers, did not suffer from the virus, were not infected by the virus, so we call them Teflons. We had access, or we have access, to 2.8 million electronic medical records. And this study, strangely enough, was carried out by Ronnie, which is infected by virus now. So she's not in Teflon, so she's not here now. This is what Ronnie did. She ran over the data sets and she looked for positive PCR infection results. Now, this is a PCR, this is not serology. We don't have serology for everybody, but we have many tests of PCR, and this was our proxy for infection. She looked at households. It doesn't have to be a family, but people that live in the same address as a household. And then we plotted that along a time interval. So out of 2.8 million individuals or electronic medical records, we came out with 252 Teflons. I'm showing you one example here. So let's look at the timeline. This is about a month and a half. We have here a 4-year-old male which underwent a PCR test, which came out negative. Okay, it's blue. Here we have his mother, 33 years old, which was positive. A day later, he was tested again and he's still negative. Okay, he's still negative here about 10 days later. And then his 8-year-old brother was found positive and he's still negative. And then he's still negative 10 days after that. So we have 252 cases, very clear, just like that, where we have 2 weeks interval test before and after. An infected person in the same family was found positive. Now, if you want to look at the different features of these Teflons, let's look at them. So in terms of males and females, we have the same amount, roughly the same amount. If we look at ages, we can see that about 25% of them are between the ages of 6 and 12. But notice this, we also have another about 25% between ages of 41 and 60, which were not infected, which live in a household with people which were infected. If we're looking here at the number of vaccinations of these 252 people, about half of them were not vaccinated at all. And you can see here a large proportion, or 25%, were about vaccinated 3 times. We're looking at a number of individuals in a family. Many of them are in households of 5 to 7 people. And we're looking here at number of positive events within that time frame of this family. We have 2, we have 3 here, and we have even more events. And of course, the question is, what makes them so unique? And I can tell you that we're still running on this data. This is the comparison here. These are permanent diagnosis. We're running very carefully on these data sets, on the clinical sets, on the medical records. We have some differences in permanent diagnosis, also in taking particular drugs. We're looking into that. We're trying to figure out. We also eventually want to add on it genetics and epigenetics, so DNA and RNA, in order to see what makes them stand out. Obviously, we can also look at the repertoire of their antibodies and figure out what makes them so unique. We like visualizing this data. Again, this is data from Shamir Medical Center, from Dr. Adina Bar Chaim's team. Here, this is between the 2nd and the 3rd vaccinations. This is after the 2nd vaccination here. On the y-axis, we have the number of days since the 2nd vaccination, and here we have the age. I think it's very clear here to notice that we have an alignment along 180 days, and above the age of 50, we have an accumulation of mortality and morbidity at this stage. We presented this together with our team to the Ministry of Health, and it was quite evident that a booster is needed at this stage in order to increase the level of antibodies. What about viral variant development? We have sequenced at Shamir Medical Center about 50,000 viral genomes. This is collapsed now to 5,000 in order to fit this graph, and you can see very clearly the development of the various variants throughout the years. This is going 2 years backwards, and you can see the appearance of the various variants throughout the time. Some of them are very divergent, and others are very similar within that branch of the tree. Going back only 6 months back, you can see very clearly shifts between variants. So here you can see the Delta variant. You can see here at the end of last year, the appearance of the Omicron, and here we can see shift from one type of Omicron to another type, and overcoming the populations. And you can see these waves coming through the different variants just by sequencing them within the population. We're not only looking at variants passing between people, but also variants within an individual. So because we use NGS deep sequencing and we could read it quite deeply, now we can see the various variants within a particular individual. If you look here on the x-axis, we're looking at allele frequency, and here we can see the density on the y-axis. On the left-hand side, we're looking at non-synonymous. On the right-hand side, we can see synonymous. So what we can see here, for example, is very low allele frequency but high density of non-synonymous mutations within a particular individual. So many mutations are non-synonymous. This is an experimental effect of evolution, of trying to generate many mutations at very low allele frequency at a particular individual. But when we're looking at those that were transmittable mutations, non-synonymous and synonymous are roughly equal in allele frequency and in the density here, meaning that those are the ones that pass from one person to the other. We're also comparing this in individuals that are immunosuppressed, and we can see a totally different map. And what we're trying to understand is what are the intra-host variations within immunosuppressed people, individuals, versus those that are non-immunosuppressed. It looks like an entirely different map. And now to the most recent viral variant, and that's variant BA1, and you can see it here on top. So the x-axis is actually the genomic coordinates, about 30,000 nucleotides from left to right of COVID-19. Here you can see the BA1 related mutations or unique mutations. On the bottom here you can see the BA2 version of the Omicron. And what we found a week and a half ago is actually a variant that looks like this. Half BA1, half BA2, cut in the center. This was from about a week and a half ago, and what we found it in 2 particular individuals. Since then, we found it in 5 additional individuals, and what we see is actually that it's not cut in the center, it shifts more towards the left. So the most recent variant from 2 or 3 days ago are those that are 30% BA1 and 70% BA2, cut in the center. When we're looking at the clinical manifestation, we see that it doesn't seem as severe. Just by going over these patients, we see that there are some structural changes and very interesting mapping it and building the 3-dimensional structures and trying to understand the effects eventually down the road. So what I showed you here, that we're generating a lot of hypotheses. We're usually coming more with more questions than answers. We're looking at the DNA now. We're trying to figure it out. We're adding pathways in the RNA. We're looking at proteins or antibodies. We're looking at a lot of electronic medical records and trying to understand those Teflons, the ones that are not infected versus infected. The person who did most of this work is Guy from my lab, a PhD student, together with Eden, who helped him. Roni did most of the Teflon study. She's not here today, as I mentioned. She's not a Teflon. She's suffering from COVID-19 now. Our collaborators is our professor, David Gurevich from the Faculty of Medicine, Dr. Abdina Bar Chaim's lab from Shamir Medical Center, and KSM Maccabi team, Dr. Sivan Gazit and Dr. Tal Patalon. Thank you. Questions? Do you still have your question?
Did you check if there was anything particular in the ACE2 of the Teflon?
In the ACE2 of the Teflons, no. We didn't. Okay. We have results for ACE2. You want to mention the analysis you did on all the ACE2 variants? It's not in the Teflons, but in the other ones.
They're supposed to at least to a degree share the same variants. So there must be more coding.
Yes.
If we can see there is more coding, we just don't know which of these hypotheses actually holds true.
So it might be a difference in global effects, but not a local effect when you're in the same household. That's the answer regarding the ACE. Yes, Ben.
Are there any other studies from abroad, different cultures in that sense, that you can cross to see something Teflons to, the Israeli population versus, other types of population?
So other labs have been looking at Teflons and trying to understand the diversity of antibodies, and they believe that this might be one of the effects. we haven't reached yet the blood of these patients. Currently, Maccabi really wants us to understand the medical records before we approach any of them and take any samples. Thank you. Next speaker. Our next speaker is